So hello students, welcome to Karnataka. This is Basraj, your biology master teacher. Students, I hope everyone is safe and sound and doing extremely well in life. Now students, today is a lecture where I'll be teaching you about microbes. Now, tell me first of all of you tell me, what comes to your mind when you think about microbes? Is it a good thing or bad thing? Every single time when you think about microbes, 99% of time, I am very sure a bad thing will come to you or a harmful thing will come to your mind. Yes or no? Bacteria, oh, dangerous. Danger. Bacteria ಅಂದ್ರೆ dangerous. ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹತ್ರ ಇರಬಾರ್ದು, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸ್ವಲ್ಪ ದೂರ ಇರಬೇಕು. ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮೈಗೆ ಬಂದ್ಬಿಟ್ರೆ ತುಂಬಾ ಕಷ್ಟ ಆಗುತ್ತೆ. That is the first thought, right? Bacteria is very bad for you. ನೀವು ನೋಡಿರ್ಬೋದು ಲೈಸೋಲ್ ಆಡ್ ತೋರಿಸ್ತಾರೆ. ಲೈಸೋಲ್ ಆಡ್ ಅಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ತುಂಬಾ ಇರುತ್ತೆ, ಕ್ಲೀನ್ ಮಾಡ್ತೀವಿ. ಡೆಟಾಲ್ ಆಡ್ ಅಲ್ಲಿ, ಲೈಫ್ ಬಾಯ್ ಆಡ್ ಅಲ್ಲಿ. So every single ad also shows bacteria in a very bad way. But students, all of you should know, everything has how many faces? Two faces. Bacteria can be good, bacteria can be bad at the same time. Okay. ನಿಮ್ಮ ಮನೆಯಲ್ಲಿ ದೋಸೆ ಮಾಡ್ತಾರೆ ಹೇಳಿ. ದೋಸೆ ಮಾಡ್ತಾ ಇರ್ತಾನೆ ದೋಸೆ ಮನೆಯಲ್ಲಿ. ಈ ದೋಸೆ ಮಾಡೋ ಪ್ರಾಸೆಸ್ ಅಲ್ಲಿ do you know there is a bacteria involved? How many of you know that? Okay. Now I'm telling you, ದೋಸೆ ಮಾಡೋದಲ್ಲಿ ದೋಸೆ ಮಾಡ್ಬೇಕಂತ ಅಂತಂದ್ರೆ ಅದರಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಆಡ್ ಮಾಡಬೇಕು. ಬಟ್ ಹ್ಯಾಸ್ ಎನಿ ಒನ್ ಆಫ್ ಯು ಎವರ್ ಅಬ್ಸರ್ವ್ಡ್ ಯುವರ್ ಮದರ್ ಆಡಿಂಗ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಟು ಇಟ್? ನೀವು ಅಷ್ಟು ನೋಡಿದಿರಿ ಯಾವತ್ತಾದ್ರು? ನಿಮ್ಮ ಮಮ್ಮಿ ಆಗಿರ್ಬೋದು, ಅಪ್ಪ ಆಗಿರ್ಬೋದು, ಯಾರಾದ್ರೂ ದೋಸೆ ಮಾಡಕ್ಕಿಂತ ಒಂದ್ ದಿವಸ ಮುಂಚೆನೆ ಹಿಟ್ಟನ್ನ ರೆಡಿ ಮಾಡ್ತಾರೆ ಹಿಟ್ಟನ್ನ. ಯಾವತ್ತಾದ್ರೂ ನೀವು ಆ ದೋಸೆ ಹಿಟ್ಟಿಗೆ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಹಾಕದ್ ನೋಡಿದೀರಾ? ಹ್ಯಾವ್ ಯು ಎವರ್ ಎನಿ ಒನ್ ಆಫ್ ಯು ಅಬ್ಸರ್ವ್ಡ್ ಇಟ್? ಐ ಆಮ್ ವೆರಿ ಶೋರ್ none of you would have observed that bacteria is being added. ಅಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆಡ್ ಮಾಡಿಲ್ಲ ಅಂತಂದ್ರೆ ಈ ಫರ್ಮೆಂಟೇಷನ್ ಆಗ್ತಿದೆ ಮತ್ತೆ. ಸೊ ಆ ಕ್ವೆಶ್ಚನ್ ನಾನು ಇವತ್ತಿನ ಲೆಕ್ಚರ್ ಅಲ್ಲಿ ಸಾಲ್ವ್ ಮಾಡಲ್ಲ. Okay, so with that being said, students, can we start today's class, that is microbes in human welfare part 1? Let's go right now, all of you. So all of you are aware that microorganisms, ಈ microorganisms ನ ನೀವು ಯಾವತ್ತಾದ್ರೂ ರಿಯಲ್ ಆಗಿ ನೋಡಬಹುದಾ ಹಂಗೆ without microscope? Can you see microorganisms without microscope? Not possible. Why? Because they are microscopic. They are microscopic and they are very tiny. ತುಂಬಾ ಚಿಕ್ಕದು. Okay. Mixing in the hand ಎಲ್ಲ ಇಲ್ಲ. Unbedi. There is hand has bacteria, hand mixing is happening. Bacteria is coming from hand. ಅಷ್ಟೊಂದಿಲ್ಲ. ಓಕೆ, ಯೋಚನೆ ಮಾಡಬೇಡಿ, ಅದು ಸೀಕ್ರೆಟ್ ಮಸಾಲಾ ಅಲ್ಲ ಅದು, ಸರಿನಾ? Besides macroscopic plants and animals, microbes are a major component of biological system on this earth. In fact, inside your stomach, inside your gut, in small intestine ಆಗಿರಬಹುದು, there are many bacteria inside your body also. Okay, good bacteria. Okay, one question in the board ಮೇಲೆ. Do you remember which kingdom among the living organisms contain microbes? ಎಲ್ಲರಿಗೂ ಗೊತ್ತು, there is 5 kingdom classification, right? Kingdom Monera, Kingdom Protista, then Kingdom Animalia, Kingdom Fungi and Kingdom Plantae. ಈ 5 kingdom classification ಅಲ್ಲಿ where do you think majority of microorganisms land? ಹೇಳ್ತೀರಾ ಯಾರಾದ್ರೂ? ಕ್ಲಾಸ್ 11 ಕಲ್ತಿರೋರು, ಕ್ಲಾಸ್ 11 tell me. ಏರ್ ಅಲ್ಲಿ ಇರುತ್ತೆ ಭೂಮಿಕಾ, ಏರ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. I'll come back to that question. Tell me. Kingdom Monera, absolutely, Kingdom Monera. So Kingdom Monera mostly has the microorganisms. Kingdom Monera ಅಂತ ಬರ್ಬಿಡೋದು ಇಲ್ಲಿ. Kingdom Monera, beautiful. Next thing is, microbes are present everywhere, ranging from soil, water, air, inside the body of animal, plants and humans. So can I tell, microbes can be even be found in the hot springs? ನೀವೆಲ್ಲರೂ ಬಯೋಟಿಕ್ ಚಾಪ್ಟರ್ ಮಾಡಿದ್ರೆ ನಿಮ್ಗೆ ಒಂದು ಗೊತ್ತಿರಬಹುದು. There is something called as thermostable bacteria. We get something called as Taq polymerase from there. Taq polymerase, PCR use ಮಾಡ್ತಾರೆ. Can anyone tell me which is that organism where we get Taq polymerase from? It is an organism which is found in the hot springs. Exactly, Thermus aquaticus. Okay, inhospitable conditions like your glaciers, geysers, thermal vents, acidic environment. Oh, Niveliga. Where do you find, which type of bacteria do you find in extreme conditions, students? Which type of bacteria do you find in very harsh condition? Do you find Eubacteria or do you find Archaebacteria? ಬೇಗ ಹೇಳಿ ನನಗೆ, ಬೇಗ ಚಾಟ್ ಎಲ್ಲ ತಿಳಿಸ್ತಾ ಇರಬೇಕು, ಬೇಗ ಆನ್ಸರ್ ಬಿಡಬೇಕು ನಿಮ್ಮ ಚಾಟ್ ಅಲ್ಲಿ. Do you find eubacteria or do you find archaebacteria in the harsh conditions? ಎಲ್ಲರಿಗೂ ಗೊತ್ತು ನಿಮ್ಗಿದು, ಎಲ್ಲರಿಗೂ ಗೊತ್ತು, ಹೇಳಿ ಬೇಗ. Now, microbes are diverse. Protozoans, bacteria, fungi, microscopic animals and plant viruses, viroids and also prions, and that are proteinaceous infecting agents, some of the microbes. So students, ಇವೆಲ್ಲ ಕೂಡ microbes. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಈ ಮೇಜರ್ ಮಿಸ್ಕನ್ಸೆಪ್ಷನ್ ಇದು. Every single time when I talk about microbes, it doesn't mean only bacteria. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ ಪಾಯಿಂಟ್ ನ. It doesn't mean only bacteria. Microbes can have something called as algae, protozoans, microscopic animals, plant viruses, viroids, ಎಲ್ಲ ಕೂಡ microbes. ಸರಿನಾ? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇಲ್ಲಿ ನಿಮ್ಗೆ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ, ಇಲ್ಲಿ ನಿಮ್ಗೊಂದು ರಾಡ್ ಶೇಪ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಸ್ಟೂಡೆಂಟ್ಸ್, what is the other name of rod shaped bacteria? Can anyone tell me? And what is the other name of spherical shaped bacteria? Rod shaped and spherical shaped bacteria has another name. For example, rod shaped bacteria is also called as bacilli. Spherical is also called as cocci. So rod shape and spherical shape is called as bacillus and cocci. Sorry, now ಇಲ್ಲಿ ನೋಡಿ, ನಿಮ್ಗೆ ಒಂದು ಫ್ಲಾಜೆಲ್ಲಾ ಕೊಟ್ಟಿದ್ದಾರೆ. It is a rod shaped bacteria right here. Okay, ಅದು ಬಿಟ್ರೆ they have given you other examples of microbes here. This right here is one main boss, ultimate boss idu. Yaake ultimate boss appa idu? This microbe right here is not infecting anything. It is infecting even a bacteria. Bacteria nu kuda safe illa. Artha ಮಾಡ್ಕೊಳ್ಳಿ, bacteria kuda safe illa, ಯೋಚನೆ ಮಾಡಿ. This right here is called as a bacteriophage, which is a virus which infects. This right here infects bacteria. Okay, so this is our ultimate boss idu. This is your bacteriophage. Then we have adenovirus which causes respiratory disorders. Then we have your TMV, tobacco mosaic virus. Tobacco mosaic virus. All of these are examples of microorganisms. Ellargu artha agtidiya? These are the different examples of microbes. Okay, now students, we can culture the microbes. ನೀವು microbes ನ ಗ್ರೋ ಮಾಡಬಹುದು. ನೀವ್ ಹೆಂಗೆ ಮನೆಯಲ್ಲಿ ಗಾರ್ಡನ್ ಇದೆ, ಪ್ಲಾಂಟ್ ನ ಬೆಳೆಸ್ತೀರಾ. ನಿಮ್ಮಲ್ಲಿ ಪೆಟ್ಸ್ ಇರುತ್ತೆ, ನಮ್ಮಲ್ಲಿ ಒಂದು ನಾಯಿ ಇದೆ, ಸರಿನಾ? ಸೊ ಐ ಗ್ರೋ ದಮ್. ಎಸ್, ಈ ತರ ಮೈಕ್ರೋಬ್ಸ್ ನು ಕೂಡ ಗ್ರೋ ಮಾಡಬಹುದು. ಬಟ್ ಫಾರ್ ದಿಸ್ ಯು ಡೋಂಟ್ ನೀಡ್ ಅ ಗಾರ್ಡನ್, ಯು ನೀಡ್ ಅ ಲ್ಯಾಬ್. ಸೊ ಎಲ್ಲಾ ಮೈಕ್ರೋಬ್ಸ್ ನಾವು ಲ್ಯಾಬ್ ಅಲ್ಲಿ ಬೆಳೆಸ್ತೀವಿ. For example, right here, this is a culture media, culture plate of a microbe. This is a culture plate of a fungi. So we can grow bacteria and fungi inside a lab. LAB, lab. Okay, inside a lab. Okay, such cultures are useful in studies of microorganisms. Okay, microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತು. There is okay, ಹೇಳಿ ಎಷ್ಟೊಂದು ಜನ ನೀವು sugar cane juice ಕುಡಿತೀರಾ ಹೇಳಿ ನನಗೆ. How many of you drink sugar cane juice? Raise your hands quickly. How many of you like to drink sugar cane juice? ನಂಗೆ ತುಂಬಾ ಇಷ್ಟ, ನಂಗೆ ತುಂಬಾ ಅಂದ್ರೆ ಇಷ್ಟ sugar cane juice. But have you noticed sometimes the sugar cane which you get near the sugar cane juice, it has some form of red color on the sugar cane? How many of you have observed it? Tell me. ನಿಜ ಹೇಳಿ ಎಲ್ಲರೂ. How many of you observed that red coloration in the sugar cane? You might have observed red color ಆಗಿರುತ್ತೆ. That is called as red rot of sugar cane. How many of you observed a plant leaf has this white powder on it? You might have observed. So all of these are plant diseases. There are wide variety of plant diseases which you need to study. Okay, now animals allu agutte. Now today's goal for today's session is going to be microbes in house products, microbes in industries. ಈ ಎರಡು ಪಾಯಿಂಟ್ ನಾವು ಇವತ್ತಿನ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಮಾಡದ. ಮುಂದಿನ ಇವತ್ತು ಇದು ಕೂಡ this also chemicals and enzymes. These 3 points we'll be studying in today's class. Next class I'll be teaching you something called as sewage treatment, biogas and biocontrol and fertilizers. 1, 2, 3, 4 in the next class, part 2. Today's class, household, industrial and chemicals and enzymes. Ready na ellaru? Ready na? So this is the thing we'll be doing in today's class. ಇನ್ನ ನಿಜ ಹೇಳ್ತಾ ಇದೀನಿ, ಈ ಚಾಪ್ಟರ್ ಇಂದ ನಿಮ್ಗೆ 99% ಆಫ್ ದ ಟೈಮ್ಸ್ ಯಾವ ತರ ಕ್ವಶನ್ ಬರುತ್ತೆ ಅಂತಂದ್ರೆ ಮ್ಯಾಚ್ ದ ಫಾಲೋವಿಂಗ್ KCET ಅಲ್ಲಿ. In board exam kuda you will get match the following. Board exam kuda nimge they will ask you which particular enzyme, give a statement. So I'm telling you again, learning the examples in this chapter is the most critical thing to do. And for the very same reason, I have a mind map for you. I'll show you. I'll give you a small glimpse of it. So all of you can download the notes and learn later on. This right here is a mind map. I think it is not visible right now. Might not be visible because it's too bright for you. But I will give it to you in the PDF format. Download the mind map and study. For example, can you see curd, bread, toddy, Swiss cheese, fungi, cyanobacteria, bacteria? Yes or no? So download this particular PDF. You will have you'll benefit from it. Okay. So with that being said, students, let's start with something called as your household products. Okay. How many of you like to eat curd? Tell me now. Ellargu ishta mosaru. Curd is amazing. Curd rice andre best curd rice ratri hottu. Tane. So how is curd made? The first thing which I'll teach you is how is curd made. Okay, ready? Now, students, you need to understand in the entire process of making curd, how does it happen in your household? You tell me. You might have observed in your house, one day before the curd which you eat, one day before in the night time, your mother will take a spoon of curd and mix it with milk, and the next day the milk is converted to curd. How many of you observed this phenomena in your house? Because it happens, right? Ella sati nobitu Nandini curd tarakagata. So costly adu. So what do? You take a small curd and you add that curd into milk, and the next day the milk is getting converted to curd. So what exactly is happening there, students? The thing which is happening here is the spoon of curd which you add, that one scoop of curd has millions of bacteria, and the bacteria which I'm talking about is none other than the lactobacillus bacteria, also called as LAB, lactobacillus bacteria, right here. An example is production of curd from milk. Microorganisms such as lactobacillus are commonly called as lactic acid bacteria, LAB, grown in the milk and converted to curd. So ಏನು ಮಾಡುತ್ತೆ? Milk ಇಂದ curd ಗೆ conversion ಮಾಡುತ್ತೆ. Which one? Lactic acid bacteria. Done. During the growth, lab produces acid that coagulates and partially digests the milk proteins. ನಿಮಗೆ I'll teach you the entire process, students. I'll teach you the entire process. Understand what exactly is happening. Understand. Very important concept idu. Very important concept. Artha madkoli. And later on, you can go tell it to your mother or anyone. How does the milk gets converted to curd? The actual process. Understand. I will not teach you the superficial. I will teach you the actual concept right now. Understand. Now, milk has many components. Milk has many components. Milk is emulsion. All of you know chemistry. It has lipids, it has water, it has proteins, and it also has a sugar molecule, a disaccharide called as lactose. How many of you heard about it? Tell me. How many of you heard about something called as lactose which is present in the milk? Some of you tell, I am lactose intolerant. You tell, right, I'm lactose intolerant. Meaning is that there is something called as lactose, there is something called as casein protein as well as whey protein. So there is lactose and there is also protein in the milk. Two types of proteins, whey protein and casein protein. All of you should know this. So what exactly is happening? When you add the curd into the milk, your bacteria, lactic acid bacteria, will attack the lactose. Lactose is broken into glucose and galactose, and this glucose and galactose undergoes a process called as glycolysis. And all of you remember glycolysis, right? Respiration in plants chapter. And the end product of glycolysis is your pyruvate or pyruvic acid. Now this pyruvate is undergoing a second step now. Link reaction ad name barutte. There is a process called as fermentation. How many of you heard about fermentation? Raise your hands. Raise your hands quickly. Raise your hands. This particular pyruvate undergoes a process called as fermentation now. Fermentation. And if you're my student, I will teach you an extra concept here. Under fermentation, there are two types: alcoholic fermentation and lactic acid fermentation. ಆಲ್ಕೋಹಾಲಿಕ್ ಫರ್ಮೆಂಟೇಷನ್ ಇಂದ ಆಲ್ಕೋಹಾಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ, ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ ಫರ್ಮೆಂಟೇಷನ್ ಇಲ್ಲ ಆಗ್ತಾ ಇದೆ. This lactic acid fermentation is occurring right here. So what is happening? I'll tell you again. Milk has lactose, and when you add your lactic acid bacteria, the LAB is broken down to pyruvate. Pyruvate is undergoing lactic acid fermentation to produce lactic acid. Illaru clear allargunu? Illaru clear? So we right now have lactic acid in the milk. Now tell me students, basic chemistry. When there is a lot of acid in the environment, will the pH increase or decrease? Kushmita, tell me. Ammu, tell me. When there is a lot of acid in the environment, will the pH increase or decrease? Tell me. If there is lactic acid, the pH will start to decrease. The pH will continuously decrease. pH is decreasing now because of lactic acid. pH is decreasing. Because of this decrease in pH, the milk starts to coagulate. The protein gets denatured. Because all of you know, proteins cannot maintain their structure in low pH. In low pH, proteins cannot maintain the structure, and the protein starts to coagulate, and that's the process. That is the process how milk is getting converted to. The proteins in the milk is getting converted to. ನೋಡಿ, ನಾನು ನಿಮಗೆ ಬರೀ ಇಷ್ಟೇ ಹೇಳ್ಬಿಡ್ಬೋದಾಗಿತ್ತು, but it's my job as a teacher to tell you the actual process. So I'll recap one more time. Recap one more time. First, we have something called as your lactic acid bacteria. It is present in your curd. Okay. So what do you do? The first step is a small amount of curd is added to fresh milk as inoculum. So the curd here is acting as a inoculum or a starter containing millions of lab, which at a suitable temperature multiplies and thus converting milk into curd. How is it converting milk into curd? Milk has something called as lactose. The lactose is broken down by the lab. When the lactose breaks down, it forms glucose and galactose disaccharide. Glucose undergoes glycolysis to produce the pyruvate. Pyruvate gets converted, undergoes fermentation. Which type? Lactic acid fermentation. As a result, in the milk we have lactose. Lactose will decrease the pH, and when the pH is decreasing, the proteins present in the milk, is converted to the curd. Now I'll tell you, ask one more question. Can lactose intolerant people, can they generally eat the curd? Yes or no? Tell me. Can lactose intolerant people eat the curd or not? See, they can't drink the milk. Milk has lactose. They cannot drink the milk. Not possible. But can they eat curd? Yes. Why? Because curd does not have lactose. The curd does not have lactose, and hence lactose intolerant people like me can also eat curd. Concept artha aita ellargunu? ಹೇಳಿ ಎಲ್ಲರಿಗೂ. Quickly fire ತೋರಿಸಿ ಒಂದು chat ಅಲ್ಲಿ if you understood the concept and not by hearting any of it. ಎಲ್ಲರೂ by heart ಮಾಡಬಹುದು ಇನ್ನ. There's nothing to learn, students. If you by heart, there is nothing difficult to by heart here. Curd, lactic acid bacteria, and converted to curd. Curd is doing what? Increasing the nutritional quality. That is increasing the vitamin B12, also called as cobalamin. But concept is important to understand. How the milk is getting converted to curd. Okay. So I hope that is understood by all of you now. The first step is over. Next is called as your idli dosa. Idli ಹೆಂಗ್ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ? idli ಹೆಂಗ್ ಮಾಡ್ತೀವಿ? How do we make dosa? Can we start now? Start ಮಾಡದ? ಈ idli dosa process ಕೂಡ, here also there is something called as lactic acid fermentation. The idli dosa batter or the dosa batter is undergoing fermentation. It is undergoing what? Fermentation. Now, the question which I asked you before, in question itto, have you seen your mother adding bacteria? No. ನಾವು ಹೇಳಿ ನಿಮಗೆ, ನೀವು ಮನೇಲಿ ದೋಸೆ ಹಿಟ್ಟು ಮಾಡಿದರೆ ನಿಮ್ಗೆ ಗೊತ್ತಿರುತ್ತೆ. ದೋಸೆ ಹಿಟ್ಟು ಮಾಡಬೇಕಾದ್ರೆ we use something called as rice. We also use something called as black gram, urud dal ನೋಡಿದಿರಲ್ಲ, ವೈಟ್ ಕಲರ್ ದಾಲ್ ಹಾಕ್ತಾರಲ್ಲ, ಆ urud dal ಅಲ್ಲಿ there is a bacteria called as leuconostoc. There's a bacteria called as leuconostoc which is present in the rice and the urud dal. And that leuconostoc bacteria is a type of your lactic acid bacteria. and that bacteria is undergoing fermentation to make the dough bigger. ಅದರಿಂದ ನಮಗೆ ಇಡ್ಲಿ ದೋಸೆ ಒಳ್ಳೆ ಫ್ರೆಶ್ ಆಗಿ ಬರುತ್ತೆ ಬೆಳಗ್ಗೆ. ಸರಿನಾ? ಡನ್. ಲ್ಯಾಕ್ಟಿಕ್ ಫರ್ಮೆಂಟೇಷನ್. ನೆಕ್ಸ್ಟ್ ಥಿಂಗ್ ಇಸ್ ಕಾಲ್ಡ್ as ಬ್ರೆಡ್ ಮೇಕಿಂಗ್. ನೀವು ಬ್ರೆಡ್ ತುಂಬಾ ಈಸಿ ಮಾಡಕ್ಕೆ, ನಿಮ್ಮ ಮನೇಲಿ ಮಾಡಬಹುದು ಬ್ರೆಡ್. The only one bacteria you need, not bacteria, you need actually a fungi, technically a fungi type, that is called as yeast. Yeast is required, that is called as Saccharomyces cerevisiae. Saccharomyces cerevisiae is required for bread making. Okay, bread making. Now toddy, yes nodi ಏನಪ್ಪ ನೀವು ಹೇಳಿ, raise your hands. How many of you seen the toddy preparation? Toddy preparation is very simple, students. What exactly happens here is, ಒಂದು ತೆಂಗಿನ ಮರ ಇರುತ್ತೆ, coconut tree. A man will climb the coconut tree and he will climb the coconut tree and he will keep a pot there. He'll keep a pot there. Now what he does is, he'll make a small cut in the leaf. Near the branch, he will cut a small cut. When the small cut is made, the sap of the coconut tree is filled into the pot. So what is sap? All of you know sap. Sap contains all the nutrients. The sap contains carbohydrates. Sap contains nutrients. So all of these nutrients are added to the pot now, to the pot. Now what happens, because of the bacteria present in the atmosphere and also his hand to some extent, the sap undergoes alcoholic fermentation to produce the toddy. You might have seen the white color alcohol in the movies, right? So that is sap undergoing alcoholic fermentation. Okay, now it is mainly done by the fermenting the sap of palms or coconut tree. Mainly palms, sometimes coconut trees. Okay, mostly palms. Okay, now there are other things also you might have seen: fermented bamboo shoots, fermented soya beans. Japan is tumba famous ido, called as natto. Okay, fermentation of curling of fish and meat, they do fermentation. So all of these are different applications. Okay. Next thing is cheese. Next thing is called as cheese. Sir, why is it hung only on the coconut trees? It is hung on the coconut trees or palm trees because they produce a lot of sap. They produce a lot of sap, and the sap can be easily be converted to alcohol. That is why. Usually if you climb the mango tree or something, the sap is not very good sap, quality of sap. Okay, now students, artha agtide tane? This chapter is very interesting chapter. Now I'll teach you how to make cheese now. Cheesing madadanta. Okay. Now students, you should know there are different types of cheese. There are different types of cheese. This mozzarella, atharadalla. There are different types of cheese only. There is cheddar cheese, there is Swiss cheese, there is Roquefort cheese. Yes, there are different types of cheese there. There are different types of cheese available in the market. And every single cheese is prepared in a different way. For example, there is something called as Tom and Jerry cheese. Yenappa Tom and Jerry cheese andre idu? This one. Have you seen this? Students, how many of you seen this cheese? Tom and Jerry cheese. Tom and Jerry cheese. And this cheese is also called as Swiss cheese. It is also called as Swiss cheese. Tom and Jerry cheese is also called as Swiss cheese. How many of you seen this in the cartoons? Tell me, raise your hands. So we want question practice series, how it is going in neat. Baratte, don't worry. Nodi, first finish the syllabus. Let's finish the syllabus first. Adadmel tane question practice baradu. If you don't finish the syllabus only, is there a point to bring the question practice now only? Nee yochne madu. Agalala. So let's finish the syllabus first and then we will do question practice. How much ever you want. Okay. So is the toddy harmful to humans? Obviously it's alcohol. Alcohol is bad for you, bad for the body. Nim liver or kidney alatte. Okay. This is called as Tom and Jerry cheese, also called as Swiss cheese. Okay, this right here, it's called as Swiss cheese. Now students, there is a bacteria here. The bacteria which is involved in making the Swiss cheese is called as Propionibacterium shermani. Repeat after me students, repeat after me. Propionibacterium shermani. Propionibacterium shermani. Repeat after me. Propionibacterium shermani is a bacteria which is used to make Swiss cheese. Now students, why are there holes here? Why do we see small holes here? Do you think the rat came and ate and went? Jerry band tindu hoita idna? Yelli, Jerry band tindu hoita idna? Illa. Then why do you think there are holes here? There are holes here because when the Swiss cheese is being formed, it is undergoes fermentation. It undergoes fermentation, that is mostly lactic acid fermentation. ಅವಾಗ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ there is a release of CO2. There is a release of CO2 as a byproduct of your cheese making. ಈ CO2 ಆಚೆ ಬರುತ್ತಲ್ಲ, when the CO2 is escaping out, it is making the small holes in the Swiss cheese. Clear up? Okay, due to release of carbon dioxide. Exactly. That is called as Propionibacterium shermani. Now students, ನನ್ನ ಜೀವನದಲ್ಲಿ ಈ ಚೀಸ್ನ ತಿನ್ನಲ್ಲ. ನನಗೆ ಎಷ್ಟಾದ್ರೂ ಕಾಸು ಕೊಡಿ, ನೀವು ಎಷ್ಟಾದ್ರೂ ರೊಕ್ಕ ಕೊಡಿ, ಎಷ್ಟಾದ್ರೂ ದುಡ್ಡು ಕೊಡಿ, ಈ ಚೀಸ್ನ ಮಾತ್ರ ನಾನು ತಿನ್ನಲ್ಲ ಲೈಫಲ್ಲಿ. ಯಾವ್ದು ಹೇಳಿ ಅದು? ದ ರಾಕ್ಫೋರ್ಟ್ ಚೀಸ್, ದ ರಾಕಿಫೋರ್ಟ್ ಚೀಸ್. ಯಾಕೆ? ವೈ ಇಸ್ ದಿಸ್ ಚೀಸ್ ಇಸ್ ನಾಟ್ ವೆರಿ ಗುಡ್ ಫ್ರಮ್ ಮೈ ಪರ್ಸ್ಪೆಕ್ಟಿವ್? ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. ಒಂದು ಚೀಸ್ ಇರುತ್ತೆ, ಆ ಚೀಸ್ ಅಲ್ಲಿ ಫಂಗಸ್ ಹಾಕ್ತಾರೆ, ಬೇಕಂತ ಹಾಕ್ತಾರೆ. So they will take a cheese and they add fungi into it. That is penicillium, not penicillium or any other fungus. Penicillium mostly is added into the cheese, and it is later it grows. The fungus grows inside the cheese, and this cheese is eaten. Apparently, I don't know. Okay, ನಾನು ತಿಂದಿಲ್ಲ ಇದನ್ನ. ನೀವು ಬೇಕಾದ್ರೆ ಟ್ರೈ ಮಾಡಿ. ಬಟ್ Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. ಯಾವ ತರ ಫ್ಲೇವರ್ ಅಂತ ಕೇಳ್ಬೇಡಿ ನಂಗೆ. It gives them a particular flavor. Okay. Now, so till now, household work ಆಯ್ತು. Till now, all of you understood? Till now, the half of the chapter is over. Today's class is over. Cheese, we have Propionibacterium shermani Swiss cheese, Roquefort cheese for your blue cheese, also called as blue cheese. Then we have your fermented toddy. Then we have bread, Saccharomyces cerevisiae. Then we have your idli dosa, that is your lactic acid bacteria. Now we'll go to industrial application. Now we'll go to something called as industrial application. Okay. Industrial application ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, in industrial application, the bacteria used in a large scale now. ನೋಡಿ, cheese and all small. Industrially they are making use of bacteria to a very large scale. ತುಂಬಾ ಯೂಸ್ ಮಾಡ್ತಾರೆ. For example, ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. In industries, microbes are used to synthesize a large number of products which are valuable to human beings. ಯಾವ ತರ? It could be your okay, ಕೊಟ್ಟಿಲ್ಲ ಬಿಡಿ, ನಾನು ಹೇಳ್ತೀನಿ. Okay, there is something like alcohol preparation ಆಗಿರಬಹುದು, antibiotics preparation ಆಗಿರಬಹುದು, chemicals preparation ಆಗಿರಬಹುದು, detergents preparation ಆಗಿರಬಹುದು, or enzymes preparation. All of these are used in the industrial application. Okay, the first thing, there are two main categories here. That is, the two common application of microbes in industries are alcoholic fermentation and manufacturing of antibiotic drugs. Alcohol fermentation ಏನಾಗುತ್ತೆ? Alcohol fermentation ಅಲ್ಲಿ we try to make something called as wine, beer, whiskey, rum. All of the alcohol thing which you have heard in the scene in the movies, heard everywhere, that is prepared with the help of microbes. Yes, all the alcohol is prepared with the help of microbes. If there was no microbes, this would have been very difficult to make. Yes, all of it is made up of microbes. Okay, now first thing you need to understand here is all the process of industrial application, it is made in large container. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. Okay, no, don't worry, Ammu, don't worry, all the notes will be given to you. Don't worry, you know, right? In the description, if you go, you will find a Telegram channel. If you go to the Telegram channel, you will find these notes tomorrow. Entire PDF will be available. Purti PDF nimge upload ಮಾಡ್ಕೊಡ್ತೀನಿ. Don't worry about screenshots or anything. Okay, and also if you want, you can join my Telegram also. There are more than 2500 ಜನ ಇದ್ದಾರೆ ಅಲ್ಲಿ, ಸರಿನಾ, ಮಕ್ಕಳಿದ್ದಾರೆ ಅಲ್ಲಿ. So this is called my Telegram channel, Baswaraj Sir Biology. This is my Telegram. Okay, BASAVARAJ. If you join here also, you'll get the notes. Don't worry, notes, you don't worry. Okay, now we have something called as fermenters. Imagine ಮಾಡಿ. Okay, ನಿಮ್ಮನಲ್ಲಿ ನಿಮ್ಮ ಮಮ್ಮಿ ಅಡಿಗೆ ಮಾಡ್ತವ್ರೆ. When your only family is there, your mother will use a small cooker. ಅದೇ ಯೋಚನೆ ಮಾಡಿ, ನಿಮ್ಮ ಮನೆಗೆ ರಿಲೇಟಿವ್ಸ್ ಬಂದ್ರು, ಇನ್ವೈಟ್ ಮಾಡಿಲ್ಲ ನೀವು, without invitation the relatives have come to your house. Now the pressure cooker will become bigger pressure cooker, ತುಂಬಾ ದೊಡ್ಡ pressure cooker. Okay, now how do they cook in weddings then? weddings ali, they have a lot of people coming in, so they use a very large container. So do you realize, based on the requirement, the size of the vessel is increasing or decreasing? More demand, bigger the vessel; less demand, small vessel. Similarly, in the case of your industries, the demand is more, so bigger vessel. See, bigger vessel is here, and that is called as fermenters. It is called as fermenters. It is a large vessel where we can control everything. ಈ ತರ ಇರುತ್ತೆ ನೋಡಿ. ನೋಡಿ, ಮನುಷ್ಯನ ಇಷ್ಟು ದೊಡ್ಡದು, ಇಷ್ಟು ದೊಡ್ಡ ಫರ್ಮೆಂಟರ್ ಇದೆ. Fermenters. They are called as your fermenters. Okay, fermenters. Now the first thing which I'll teach you something called as how to make alcohol. Okay, how to make alcohol. First, they need a raw material. The raw material varies. It can be in the wine, it is usually the grapes. In whiskey, rum, there is usually your wheat and barley. Same goes for almost for beer also, right? The raw material changes based on the alcohol, but the entire process remains the same. Okay. Microbes, especially yeast, has been used from time to time immemorable for the production of beverages like your wine, whiskey, beer, brandy and rum. For this purpose, same yeast, that is Saccharomyces cerevisiae, bread martivora, bread, same yeast is used. Saccharomyces cerevisiae, that is used for bread making, is used for fermenting malting cereals and fruit juices to produce ethanol, that hence also called as brewer's yeast. Here it is called as brewer's yeast. In bread making, it is called as baker's yeast. Ali bakery, it is brewery. Bakery, brewery, difference. Okay, depending on the type of raw material used for fermentation and types of process with or without distillation, different types of alcohols are produced. Okay, so beer and wine are non-distillated. There is water content is more. When compared to whiskey, brandy and rum, it is more distillation. Distillation means most of the water has been evaporated here. Most of the water has been evaporated. Okay, that is called as distillation. It is the entire process there. Okay, fractional distillation and all. Okay, now that was your alcohol. The next category of your products is going to be your antibiotics. Antibiotics. All of you know antibiotics. All of you have taken antibiotics. Now students, how do you think the antibiotics came into existence? The first antibiotics ever bantu world alli. I'll teach you that also now. Okay, now first of all, understand what is antibiotics. Anti means against, biotic means life. It is against life, a chemical substance which are produced by some microbes. So what is the source of your antibiotics? The source of your antibiotics is microbes that can kill or retard the growth of other disease causing microbes. Okay, now students, understand one kate heltini keliskoli. One kate, there was a scientist called as Alexander Fleming. What he did was, when he was working on Streptococcus, when he was working on a bacteria, he was working with a bacteria. Okay, now what he did was, one day he did not clean the plates and he left the infected bacteria culture plate, a plate itala, in the sink alange biputta. We also do it. Sometimes we don't wash the plate, sometimes we wash the plate, right? There is a very high chance that in your life, sometimes you have just kept the plate somewhere and you don't wash it for a very long time. Okay, so when you leave the plate for a very long time, bacteria grows there very well. But Alexander Fleming noticed that in one particular spot, bacteria was not growing there. In fact, bacteria could not grow there, or rather bacteria tried to grow and it died there. Why? Because in that very spot there was a fungi growing there called as Penicillium notatum, and this Penicillium notatum fungi has antimicrobial activity and it was killing all the microbes around it. I'll show you image. You get a better understanding. See here, this is an entire plate. Here we have penicillium. Can you see in this spot here, in this spot right here, there is no microbes growing in this spot. Why? Because of the penicillium. It is producing antibiotics. Okay, antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of others. Okay, penicillium. Okay, discovery was accidental discovery. Alexander Fleming was working on Staphylococcus. Staphylococcus bacteria, or Staphylococcus bacteria, once observed that mold growing in one of his unwashed culture plates around which Staphylococcus could not grow. He found that it was due to the chemical produced by the mold and named it penicillium after the mold Penicillium notatum. Okay, so Penicillium notatum is used as a antibiotic right now. Antibiotic. Okay, and later on, there were two other scientists who did a proper research on it. That is basically the Ernest Chain and Howard Florey. So Chain and Florey are the two scientists who worked on it. However, its full potential as an effective antibiotic was established much later by Chain and Florey. This antibiotic was extensively used to treat American soldiers in the World War II. Fleming, Chain and Florey were awarded the Nobel Prize in 1945 for the discovery. Okay, 1945 for the discovery. Now, very disease agirbodu, not just that simple disease. Antibiotics are used to cure many diseases. It could be like, Kali Kasi, that is basically whooping cough. Diphtheria, that is the Galgotu. Then there is leprosy, that is Kushrog. These are like Hindi names. Remember leprosy, diphtheria, and whooping cough. Okay, which used to kill millions of go over the globe. Munche. Now nothing is happening because of the alcohol, all of you know, that is Saccharomyces, and enzymes. These two topics I'll teach you right now. Okay. Now, first example, organic acids. There are multiple types of acid: acetic acid, citric acid, lactic acid, and butyric acid. All the different types of acids are produced by different types of enzymes, different types of microbes. Whooping cough is basically like, you cannot stop coughing and all. Okay. Sir, give the mind map, please. I'll give you the mind cap. Don't worry, I'll give. Students, knowledge na nanu share ಮಾಡೋದಕ್ಕೆ ಬಂದಿರೋದಿಲ್ಲಿ. What is the point of me keeping it? It will be only helpful if I share the mind map, right? Okay. So acetic acid is produced by your Acetobacter aceti. Citric acid is produced by Aspergillus niger, which is a fungi. Bacteria, fungi. Lactic acid is by Lactobacillus, and butyric acid by Clostridium butylicum or Butycilum. Okay, these are the different acids. These are different microbes producing the organic acid. Okay, so idna ನೀವು ನೆನಪಿಕೊಂಡು ಬರೆದು ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡಬೇಕು. Otherwise very difficult to remember. ನೋಡಿ, ಇದು ಅರ್ಥ ಆಗುತ್ತಲ್ಲ? Acetic acid, Acetobacter aceti, same meaning. This one ಸ್ವಲ್ಪ different. Remember it as AC. Aspergillus niger, citric acid. Lactobacillus, lactic acid. Clostridium butylicum, butyric acid. So these three are easy to remember. This one you need to remember extra separately. Okay. Now, Acetobacter aceti, fermented alcohol which helps in Acetobacter. This is your Aspergillus niger, nodi. Fungi. Citric acid. Clostridium, Lactobacillus. Both of you know. This one you know already, lactic acid. Okay. Now, alcohol, Saccharomyces cerevisiae. That is brewer's yeast. Okay. Next thing is different type of enzymes. Microbes also are used to production of enzymes. All of you know what is enzyme? Yes, Lactobacillus is also used to convert milk into curd, even for dosa also. Okay. All of you know what is enzyme? Enzyme is a biocatalyst which can alter the rate of reaction. Yes, biomolecules chapter. Okay. Enzymes are biological compounds, proteinaceous in nature, that catalyze the biological process. There is proteases, pectinases, streptokinases, lipases. All of them are very important, and also the function is very important. For example, first one is called as lipases. Lipases are used in detergents, formulations, and helpful in removing oily stains. That is lipases or lipases. Lipids, making better lipids. Then we have pectinases, proteinases. ನೋಟಿಸ್ ಮಾಡಿರ್ಬೋದು ನಿಮ್ಮ ಮನೆಯಲ್ಲಿ ಮ್ಯಾಂಗೋ ಜ್ಯೂಸ್ನ ಮಾಡಿ ಅದೇ ಜ್ಯೂಸ್ನ ನೀವು ಅಂಗಡಿಯಿಂದ ತಗೊಂಬಂದ್ರೆ the fruity, tell me, the fruit is more clear or watery, or the juice which you make in your house is more clear? Tell me, which one is more clear? The juice which you get from the shop, that is basically the fruity and Tropicana, is that more clear, or the juice which you make in the house? The juice which you make in the house has a lot of fibers. It has sclerenchyma. It has proteins in it, more proteins. But the same juice which you get from the market, that is Tropicana, fruity, that is more clear. Why? That is more clear because of the enzyme present in it. A fruity madbekadre, they add an enzyme called as proteinases and pectinases. The bottled juice, fruit juice bought to the market are clearer are those compared to the made at home. This is because bottled juice are clarified by using the pectinases and proteases. Done. Then what is streptokinase, students? Streptokinases. Streptokinases produce something called as a streptokinase. By it is streptokinase is produced by streptococcus. It is produced by streptococcus. Whenever this is the arteries, this is the blood vessel. In the blood vessel, there is a very high chance that sometimes what happens is there is clot formation. There is clot formation like this. And every single time there's a clot formation like this clot can be burst open with the help of streptokinase. So streptokinase is helping what? It is helping in clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to the heart attack. Okay, so streptokinase, clot buster, pectinase proteases, fruit juice, lipases, detergent. Now students, a joke ಕೇಳಿದಿರಲ್ಲ ನೀವು. If I have to buy iPhone, what do I have to sell, students? For example, today I want to buy an iPhone. I have to sell my kidney for it. If I have to buy a iPhone, I have to sell my kidney. Okay, I sold my kidney. Now later on I get to know that I need one more kidney. Okay, one kidney is gone, so I need one extra kidney from my sister. For example, my sister is giving me her one kidney to me. But to make sure the kidney doesn't get rejected from my body, to make sure the graft processing, okay, because of graft reaction should not happen, we suppress our immunity to make sure the other kidney is not being affected. Sarina, artha madkoli, whenever the patient is undergoing organ transplant, when a patient is undergoing organ transplant, we decrease the immunity of that person to make sure that the new organ is not rejected by the body. Bere kidney inda nan kidney hakak nodak hodre, there is a very high chance that my body will reject that kidney and my cells will attack it. To suppress that, we use immunosuppressant. And that's one of the amazing source of immunosuppressant is coming from your cyclosporine. That is, cyclosporine is a immunosuppressive agent produced by the fermentive action of fungus Trichoderma polysporum. So Trichoderma polysporum is a fungi. Trichoderma polysporum is a fungi which is producing what? It is producing cyclosporin A. Very, very important point. The most important point here. Trichoderma polysporum is a fungi which is producing cyclosporin A, which is acting as a immunosuppressive agent. Okay, cyclosporin A. Now, apart from cyclosporin A, the last thing which you will remember is something called as statins. Statins are blood cholesterol lowering agent produced by the fermentative action of yeast, that is Monaskis purpurus. Monaskis purpurus is a yeast which is producing statins. And statins will do what? Lower the blood cholesterol level. That's all for today's class, students. That's all for today's class. I hope if this is not visible, to all of you, I will read it. The PDF will be available. You can enjoy from there. First of all, let me read for you, some of you. Here we have the microbes in household, Swiss cheese, Propionibacterium shermani, bread, Saccharomyces cerevisiae, curd, lactic acid bacteria. Done. Microbes in industry, fermentive, again for making alcohol, Saccharomyces cerevisiae, antibiotic, Penicillium notatum, organic acid, Aspergillus niger, citric acid, Acetobacter aceti, acetic acid, Clostridium botulinum, butyric acid, Lactobacillus, lactic acid. Enzymes, lipases, pectinases and proteases, streptokinases from streptococcus, used as a clot buster. Bioactive molecules, statins from Monaskis purpurus, used as blood cholesterol lowering agent. Cyclosporin A from Trichoderma polysporum, used as immunosuppressive agent. So with the help of this mind map, you can remember so many different types of examples which will help you. Clear allarguno? So with that being said, students, I will share the entire session with you. Entire PDF will be available. Thank you so much for today's class. I will see you in the next video. ಲೈಕ್ ಮಾಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಕಮೆಂಟ್ ಅಲ್ಲಿ ತಿಳಿಸಿ ನಂಗೆ, ಕಮೆಂಟ್ ಅಲ್ಲಿ ತಿಳಿಸಿ ಹೌ ವಾಸ್ ದ ಕ್ಲಾಸ್ ಟುಡೇ. ಕಮೆಂಟ್ ಅಲ್ಲಿ ತಿಳಿಸಿ ನಂಗೆ ಹೌ ವಾಸ್ ದ ಕ್ಲಾಸ್. ವಿಡಿಯೋನ ಶೇರ್ ಮಾಡಿ, ಬಿಕಾಸ್ ದೇರ್ ಆರ್ ವೆರಿ ಫ್ಯೂ ಪೀಪಲ್ ಹೂ ಆರ್ ಟೀಚಿಂಗ್ ಇನ್ ಕನ್ನಡ ಅಟ್ ಹೈಯರ್ ಲೆವೆಲ್. Okay, so thank you so much students. This is Baswaraj sir, your biology master teacher, and those of you who do not know me, I also teach for NEET category, that is mostly botany for the NEET category students for the past 2.5 years. Okay, so thank you so much. Bye bye, take care all of you, have an amazing night. Malkoli hogi odkoli onsati, part 2 next week baratte. Okay, so bye bye, take care students. I will see you in the next video. Have a great day ahead all of you.